Hi, welcome to my presentation on Chapter 13, Microbial Evolution and Systematics. So first we'll go over a very brief history of the origin of life and some of the major events in early microbial life on Earth before we get into evolution and systematics. So we'll start with the hypothesized origins of life. So of course no one really knows how life began or where, and there are different hypotheses. But one of the leading hypotheses states that life began near deep sea vents. And we've done experiments that show that under the chemical conditions and physical conditions that we think existed in the early Earth near these deep sea vents, it would have been possible to have amino acids form and lipids and even nucleotides form non-biologically or by abiogenesis. So just due to physical processes and with the chemicals that were available in the ocean and coming up through the vents at that time, you could have formed amino acids and lipids. Actually, we haven't shown the full formation of nucleotides yet, but we've shown formation of their pieces. So we've shown formation of ribose and formation of the nitrogenous bases, and then of course you have phosphate too. So all the pieces that we need for a nucleotide would have been there. In these deep sea vents, you have these large mineral structures forming that have teensy little pores in them or little bubbles in the rock. And we believe those little compartments may have acted like early cells. So some of the molecules inside of the cell that we would normally find inside of the cell would have come about before the actual cell membrane and before you had a proper cell. But you still need some type of a compartment to contain all these molecules and let them work together. So these little tiny bubbles in the rock might have served that role at first. We also think that RNA was the first macromolecule to evolve before polysaccharides or large lipids or DNA or protein. So you have a hypothesis called the RNA world hypothesis, which you may or may not have heard of before. That's just the idea that RNA comes before DNA and protein, or RNA arose before DNA and protein. And the first organisms used RNA for the jobs that are now done by DNA and protein, which for DNA is storing genetic information and for protein is doing work. So RNA has a sequence of nucleotides, which means it stores information, so it can do the same job as DNA. RNA can also form into complex 3D shapes, which allows it to do catalysis, which normally is a job done by proteins. So it can store information like DNA, and it can do catalysis like proteins. And so it was most likely the first molecule to do both of those jobs. And I've got organisms in quotes there because at this time we wouldn't be talking about something that we would recognize as a cell. It's not a proper cell because it doesn't have a membrane. It would just be a little pore in this rock that has RNA inside of it and some other molecules inside of it. So after the development of RNA, at some point after that, you would have had protein arise and then DNA. DNA is a more stable molecule than RNA, so it's actually better at storing information for a long time. So DNA would have replaced RNA at some point as the molecule of heredity. Protein can form more diverse shapes, more diverse complicated shapes than RNA can. So once protein arose, it would have at some point replaced RNA as the main molecule of catalysis as well. And then you're just left with RNA, like for instance, our RNA in the ribosome doing the work of, most of the work of translation and serving a few other roles in the cell. At some point, you would also have the development of a cell membrane, with lipids basically spontaneously assembling to form that membrane, we think. So actually, lipids do spontaneously assemble into these little circular structures or spherical structures. If you have just one layer of lipids, that will spontaneously assemble into a micelle, a little bubble basically, like a soap bubble. A soap bubble is actually a type of micelle. If you have two layers of lipids, they can spontaneously form a lipid bilayer, which is what the cell membrane is. So probably you would have had protein and DNA arise first, replace RNA, and then at some point after that, you would have had lipids assemble into cell membranes, and then you have like actual cells. And you don't need these little rock pores anymore to contain the molecules of life. During this very early time in the genesis of life, we think that genes would have been exchanged very freely between different cells, which is called horizontal gene transfer. So you would have had a lot of these genes transferring horizontally between cells that aren't necessarily related to each other by heredity. At some point after that period, you would have had Luca, the last universal common ancestor, and from Luca, all other life came about. But of course, Eukarya did not evolve at first. Soon after Luca, you would have had bacteria and archaea evolve from Luca, and then archaea would have evolved. Eukarya would have evolved later on. One of the most important events in the early Earth's history, apart from the origin of life itself, is the Great Oxidation event, which happened sometime after cyanobacteria evolved, were the first to evolve oxygenic photosynthesis. So photosynthesis had evolved by this time already in different types of bacteria, but it was anoxygenic photosynthesis, which means that it does not create oxygen. Cyanobacteria were the first organisms that did oxygenic photosynthesis that does create oxygen. So at that point, the amount of oxygen in the earth started to rise. The Great Oxidation event actually refers to the oxidation of the Earth's atmosphere due to rising levels of O2. However, this did not happen right after the cyanobacteria evolved. It took a long time, actually. You can see that in this timeline on the side here. So right around here, 3.5 billion years ago, well, actually closer to like 3.4, you have the first anoxygenic photosynthesis evolving. And then maybe more like 2.9, 2.8 is when cyanobacteria evolved, and with them oxygenic photosynthesis. So at this point you have a significant production of oxygen beginning. However, this red line is showing the amount of oxygen in the atmosphere. So you can see that when the cyanobacteria evolved, you still had essentially no oxygen in the atmosphere. And that persisted for hundreds of millions of years, actually. Starting around 2.5 billion years ago is when you have oxygen levels finally start rising in the atmosphere, and the beginning of the Great Oxidation event. So what was happening in the meantime is just that all of this oxygen that the cyanobacteria were producing was remaining in the oceans. We believe that the early Earth's oceans actually were full of dissolved iron. So they would have had like a red color or a brownish color. They wouldn't have been blue because they had so much iron in them. So when these cyanobacteria first started producing oxygen, that oxygen would react with the iron, basically causing it to form rust or iron oxides. And then that rust would have fallen to the bottom of the ocean, and over the course of hundreds of millions of years, you would have had all that iron in the ocean react with oxygen and precipitate out, falling to the bottom, which would eventually leave you with a clear blue iron-free ocean, except of course for the bottom. And at that point, the oxygen that the cyanobacteria were continuing to make would actually be able to escape the ocean and go into the atmosphere, and you would have had levels of oxygen in the atmosphere starting to rise, which caused the Great Oxidation event. The Great Oxidation event was good for some organisms and bad for others. Overall, it was a good thing for sure, but you did have organisms that were not adapted to survive in the presence of oxygen. because as we mentioned in last week's chapter, oxygen can form reactive oxygen species. So there's a number of enzymes that will kind of cause that to happen as like a side effect of their operation. But light will also actually cause the formation of reactive oxygen species from oxygen, from O2. So O2 itself is not toxic, but reactive oxygen species are toxic because they're so reactive. So all these organisms that were living exposed to the atmosphere that were not prepared to deal with these reactive oxygen species would have died off in any environment that was exposed to the air, which would at this point have a lot of oxygen in it after the Great Oxidation event. So all of your anaerobes would have been dying off from these surface environments and from the very top of the water bodies. They would have only been able to continue living sort of deeper in the ground, deeper in the ocean, in places where you still wouldn't have had a significant amount of oxygen or any at all. Of course, the Great Oxidation event also allowed the evolution of aerobic respiration, which is the most efficient form of respiration possible that lets you make the most ATP possible. So this allowed these organisms that evolved this form of respiration to make more energy using the same amount of resources as everyone else at that time, which gave them a big advantage. So aerobic respiration spread and became very common amongst microbes, and eventually, along down the line, allowed the development of multicellular life. And the last important thing that happened as a result of the Great Oxidation event is the formation of the ozone layer. Ozone is O3. So when light hits O2, or just molecular oxygen, it does cause the formation of reactive oxygen species, but it also causes the formation of ozone, or O3. And then O3 absorbs UV rays very well. So in this image right here, you can see the ozone layer very high in the atmosphere, and you have the different types of UV rays coming down from the sun into the atmosphere: UVA, UVB, and UVC. So you can see that the UVC rays are entirely blocked by the ozone layer. UVB rays are almost all blocked by the ozone layer, only a little bit gets through, and UVA rays, most of them get through, only a few of them are blocked. So the ozone layer is dramatically cutting down on the amount of UV rays that are actually reaching the surface of the planet where life is found. And actually UVA rays are the least dangerous because they have the least energy. UVC rays are actually quite dangerous because they are very energetic, and they're completely blocked by the ozone layer. So this is very important for all life forms that live exposed to light, basically. Before the development of the ozone layer, in order to survive just on the surface of the Earth without anything concealing you from the UV light, you would need to basically be radiation resistant. Because that UV light causes a lot of damage to DNA, causes a lot of mutations, and also just basic DNA damage. So the ozone layer is protecting us from that damage and from those mutations. So we'll again briefly go over the universal tree of life, which is basically like the family tree of all life, or is attempting to be the family tree of all life. Of course, it's not complete yet. It's based on evolutionary relationships between organisms, which means that it is a phylogenetic tree. A phylogenetic tree is showing relationships between organisms based on evolution. So it's not related to physical characteristics. We have based the universal tree of life on genetics, on comparing the genetics of different organisms. Based on the RNA gene sequences, we are able to divide life into 3 domains: bacteria, archaea, and eukarya. So Luca lived soon after the origin of life, and soon after Luca lived, you had life divide into bacteria and archaea. And then at some point after that, eukarya branched off of archaea, most likely. So that's all based on the rRNA genes. There's also other genes that you can find in almost every species living today. And we believe that those genes have come from LUCA. So those are 60 highly conserved genes that we believe come from LUCA. A highly conserved gene is basically just a gene that doesn't change much through time, so it doesn't mutate very much. In general, that means that this gene is very important for the function of the organism, so that if you have a mutation in it, it's likely that the organism will die. And a final point to mention here is horizontal gene transfer, or genetic exchange between organisms that's not part of heredity. That has happened a lot, especially for prokaryotes and especially early in the history of life. And it really makes it difficult to tell where genes have come from. So normally you would like to assume that if two organisms have the same gene, they got it from a common ancestor. Right, and that would be why all organisms basically that we know of have these 60 conserved genes. They all come from this common ancestor, which is LUCA. But there's some genes that can actually be transferred not by heredity from a mother cell to a daughter cell, but horizontally. There's 3 major mechanisms of horizontal gene transfer, which we'll actually briefly mention later on in this chapter. But basically, that would be like exchange of genes between organisms that are not related to each other by common descent, except in the sense that, you know, all life is related. And so that makes it difficult in some cases to tell if you have these 2 genes, or if you have one gene in 2 different organisms, did that come from heredity or did that come from horizontal gene transfer? And then that makes it difficult to build an accurate universal tree of life. So when we try to build this tree, we try to avoid using genes that we think were transferred by horizontal gene transfer. So we had already gone over endosymbiotic theory in chapter 2, but we'll come back to it again here. Endosymbiotic theory just states that mitochondria and chloroplasts used to be independently living prokaryotes, bacteria specifically, and at some point they were engulfed by an early eukaryote, and rather than dying there, they multiplied and eventually became an organelle. This would have been a symbiotic relationship where the host cell, the eukaryotic cell, is getting free energy, free ATP from the mitochondria. From the chloroplast, it would have been getting free ATP and also free glucose, or free material to build cellular structures. So the host cell obviously benefits. The bacterial cell that was engulfed also would have benefited because now it's living in a very stable environment. It doesn't have to worry about, you know, changing pH, changing salt levels, nutrients not being available. All of that is going to be provided for it by the cell that is living inside of. And some of the evidence for the endosymbiotic theory, again, we'll just run over it quite quickly, are that these organelles, the mitochondria and the chloroplasts, have 2 membranes around them, which is consistent with endocytosis, which is the mechanism that cells use to take in large particles or large objects, wrapping it basically in a little piece of their own membrane, and then putting it into a vesicle. Mitochondria and chloroplasts also have their own genomes, their own DNA genomes, and those genomes look more like bacterial genomes than like eukaryotic genomes. So they are circular. Bacteria have a circular genome, eukaryotes have a linear genome. Mitochondria and chloroplasts have a circular genome like bacteria do. And there's also other things about the genome that makes it look more like it belongs to bacteria than like it belongs to a eukaryote. Although I will have to add that a lot of the genes that are needed by mitochondria and chloroplasts do exist in the nucleus, which means that if the endosymbiotic theory is correct, these genes would have had to initially be inside of the mitochondria or inside of the chloroplast, and then at some point they would have had to been transferred into the nucleus. And it is difficult to see how that could have happened. But also the mitochondria and chloroplasts have their own ribosomes, so they're able to make their own protein. No other organelle has that characteristic, and they divide independently on their own schedule, basically. If you look at their rRNA, it looks like bacterial rRNA. It does not look like eukaryotic rRNA. And you're actually able to match their DNA with specific types of bacteria. So mitochondria, if you analyze their DNA, it matches up with the phylum Proteobacteria. For chloroplasts, if you analyze their DNA, it matches up with the DNA of cyanobacteria. So this diagram is showing the basic outline of endosymbiosis. So you would have had, at some point, the formation of this early eukaryote. There's different, I guess, variations of endosymbiotic theory. In some of them you start out with a eukaryote that engulfs a bacteria. In some of them you start out with an archaea that engulfs a bacteria and then becomes a eukaryote, which is what's shown right here. We don't really know which way it happened, of course. But this early bacteria that's engulfed by this larger cell is going to develop into a mitochondrion. That begins dividing according to its own schedule, so you end up with a lot of those little mitochondria in this cell. Some of these cells would have evolved into animal cells or fungal cells. Some of them would have evolved into plant cells after you had the second endosymbiotic event, in which this early eukaryote engulfed a cyanobacterial cell that then became a chloroplast. So the last major event in microbial life to talk about is the origin of the domain Eukarya, or the origin of eukaryotes. We're not really sure where eukaryotes come from or how they evolved. It's one of the big questions in the early evolution of life. And it's hard to answer because eukaryotes are chimeras, which means that parts of them come from one place and parts of them come from a different place. They're kind of a hodgepodge, or like a mosaic. So some of their genes are like bacteria, some of their genes are like archaea. Their genes for working with their DNA or for gene expression are like archaeal genes. That would include the genes for transcription and translation. But their genes that are more for basic metabolism or basic catabolism and anabolism, those are more like bacterial genes. So their genes for respiration or for fermentation, those look like the same genes in bacteria. Also, the membrane of the eukaryote is like a bacterial membrane. Both the eukaryotes and bacteria have membranes made of phospholipids. Archaea do not have phospholipids in their membrane. They have different types of lipids. So it's like parts of them are from archaea and other parts of them are from bacteria, which makes it hard to see how they would have evolved exactly or when. But overall, they look more like archaea than like bacteria, which is why when we draw the universal tree of life, we always show eukarya branching off of archaea. And we'll go over a couple different hypotheses for how this might have happened. First, you have the serial endosymbiosis hypothesis, which states that first you started with an archaea, it evolved a nucleus. At that point, it's a eukaryote since it has a nucleus. And then later on, you would have had mitochondria acquired by endosymbiosis, and then chloroplasts. So that's illustrated in this diagram here. Down here, you would have Leuca. Leuca then split into bacteria and archaea, or I should say the lineage of Leuca split into bacteria and archaea. Then one of these early archaeal cells at some point evolved a nucleus, which makes it a eukaryote. So then you would have eukarya branch off of from archaea. Then some point after that, you would have had these proteobacteria be engulfed by this early eukaryote, and it becomes a mitochondrion. Then at some point after that, you would have had this cyanobacteria be engulfed by an early eukaryote and become a chloroplast. So this theory is pretty good, or not a theory, actually a hypothesis. This hypothesis is pretty decent, but there's one major thing that it doesn't explain. It does not explain why eukaryotes have a membrane that's like bacteria. If eukarya just come from archaea simply like that, it's just an archaea that evolved a nucleus, then you would expect the membrane to look like an archaeal membrane, not like a bacterial membrane. So that's strange. Then you have the symbiogenesis hypothesis, which states that you had an early archaeal cell in a symbiotic relationship with an early bacterial cell, and eventually that archaea cell engulfed the bacteria, and the bacteria became a mitochondrion, and the archaeal cell evolved the nucleus. And at that point, it was a eukaryote. And then later on, you had a cyanobacteria engulfed by eukaryote, which gave rise to chloroplasts. So that's illustrated in this diagram here. So here you have your early archaeal cell in a symbiotic relationship with an early bacterial cell. So they're kind of living in close association with each other, and each of them is helping the other. And then at some point, this archaeal cell engulfs the bacterial cell and evolves a nucleus. And at that point, it is the first eukaryote, and it already has a mitochondrion inside of it. And then later on, another eukaryote would have engulfed a cyanobacteria, which would have formed a chloroplast. So in this hypothesis, one more detail of the hypothesis is that the bacterial genes that code for the enzymes that build lipids in the membrane would have been transferred from the bacteria to the archaea after the bacteria was engulfed. And then those genes would have taken over the job of building the membrane, which would be why the membrane looks like a bacterial membrane and not like an archaeal membrane. Right, so once this bacteria is engulfed into the archaeal cell, the genes for building a membrane, basically, that are inside of this bacteria would have been transferred to the archaeal cell. They would have been transferred into the nucleus after it formed that nucleus. And at that point, those genes are what's going to be used to build the membrane, and that's why the membrane of the eukaryote looks like the membrane of the bacteria. So again, we don't really know which of these is correct, or maybe neither is correct, and it's a different sequence of events that happened. These are just two possibilities. Okay, so now we'll go over microbial evolution, starting with just evolution itself, and then we'll get into things that are special about how microbes evolve compared to non-microbes. So there's a few different ways to define evolution. A simple definition is that evolution is a change in characteristics of a population over time. A more complex definition is that evolution is a change in allele frequencies of a population over time. So you may remember from 1306 that an allele is a version of a gene. So for instance, in flowers, if you have flowers that are the same type but they're different colors, like different colored roses, for instance, you're going to have a gene in the rose that is for the color of the rose. And if you have a white rose, that means you have a white allele for that gene. If you have a red rose, that means there's a red allele for that gene. Then of course there's like pink and yellow and orange too. So all of those are going to be different alleles in the gene for a basic type of gene. There's also situations where it can be more complicated than that, but that's a basic situation. Then the allele frequency is what percentage of that gene in the population is accounted for by that specific allele compared to other alleles that exist for the same gene in the same population. So this is kind of a graphical representation of allele frequency changing over time. First you have this generation of parents. These would be pea plants, and they have flowers that can be either purple or white. So they have two different alleles for the flower color gene, purple and white. Purple is dominant, and we show that with a capital W, or we symbolize that with a capital W. The white allele is not dominant, so it's recessive, so we symbolize that with a lowercase w. If you have a pea plant that has both alleles, that has a purple allele and a white allele, the dominant one is going to win, which is the purple allele, so that gives you a purple flower. If you have two purple alleles, you get a purple flower. Then if you have two white alleles, you would get a white flower. So to determine the allele frequency in this population of parents, you're going to count up every capital W and then every lowercase w and compare the ratio of them. So for capital W, for the purple allele, you have 13 W's here, out of 18 alleles total, which gives you a frequency of 0.72, or 72% of all alleles for this gene in this population are the purple allele. For the white allele, you have 5 of them in a total population of 18 alleles, so that gives you a frequency of 0.28. So these parent generation flowers or plants are all going to kind of breed with each other and have offspring that form the new generation's population. If you look at the allele frequency in the new generation, it's going to be different than from the parent generation. So if you were to count that up, all the purple alleles, you would have 8 purple alleles now out of 18, which gives you a frequency of 0.44. And you have now 10 little white alleles out of 18, which gives you a frequency of 0.56. So that means that the allele frequency has changed over time here. For the purple allele, it's changed from 0.72 to 0.44, and for the white allele, it's changed from 0.28 to 0.56. So that means that technically you've had evolution occur here. And this is basically inevitable with every new generation. You're just going to have small changes like that, and sometimes larger changes as well. That change in allele frequency, or that change in characteristics as a result of the change in allele frequency, is evolution. So for microbes, this isn't an exact translation because most microbes are haploid, which means they only have one copy of every allele. They're not getting a copy of their genes from the mother and another copy from the father. They just have one copy from the parent cell because they reproduce asexually. So your prokaryotic microbes, the bacteria and the archaea, wouldn't have like WW, they would just have one W. So they're either going to have the purple allele or the white allele, but they could not have both. That actually means that the allele frequencies for those prokaryotic microbes are going to change more quickly than for the eukaryotic microbes that have that are diploid and have 2 copies or 2 alleles for every gene, or 2 copies of every gene. One from, well, I guess actually for microbial eukaryotes, they would also be reproducing by asexual reproduction, but they still have their 2 copies. In order to have evolution, you need to be able to have new alleles. If you only had one allele for every gene, or one version of every gene, you're not going to have evolution. It's impossible. So that means you need some source of new alleles, or a source of genetic diversity. Those sources are mutation and recombination. We'll talk about those in a moment. And you also need to have some type of process that is driving the evolution to occur. 2 of the main processes are natural selection and genetic drift. You also have some other processes, like there's artificial selection. That would be like, if you're breeding dogs, that is artificial selection. You also have sexual selection, where organisms are choosing who to mate with, but that does not apply to microbes because they reproduce asexually. So we'll go over the sources of genetic diversity, or the sources of new alleles. First, you have mutation, which is a change in the sequence of DNA that is passed down to offspring, which means that it's heritable. So if you have some type of a change in your genetic sequence and then your children end up with the same change, that's a mutation that is heritable. Most mutations are going to come from replication mistakes. So when DNA pol 3 accidentally puts the wrong nucleotide, that can lead to a mutation if the mistake is not fixed. So most of those mistakes will be fixed during proofreading, and cells will also have other mechanisms to fix those types of mistakes, but none of those mechanisms are going to be perfect, which means that you're always going to make some mistakes, or DNA pol 3 is always going to make some mistakes, put in some wrong nucleotides, and some of those mistakes are not going to be fixed in proofreading. Some of them are going to get through and not be fixed by any mechanism and end up as mutations that are permanent and that are passed on to daughter cells. You have a lot of different types of mutations, actually. One of the most interesting is a duplication mutation. A duplication mutation involves copying a gene It actually could also be just a part of a gene, or it could be multiple genes. And then the new copy is free to evolve without affecting the function of the first copy. So here's a diagram of a duplication mutation using a eukaryotic chromosome, and you can tell because it's linear, it's not a circle. Here on this chromosome you have gene A and gene B. After the duplication mutation, you're going to have an extra copy of gene A and an extra copy of gene B added to this chromosome. So now, if the new copy of gene A gets a mutation that causes it to lose its function, that's actually okay. The cell will be fine because it still has the original copy of gene A that is still functioning normally. So as long as the original copies maintain their original function, the new copies can mutate freely, they can lose their function, it's not a problem, it doesn't affect the cell, and they're going to accumulate more and more mutations as time goes by. They may eventually actually gain a new function. And you actually have a lot of genes that are like that had been copied in a duplication mutation, and since then they've had a lot of mutations occur, and they've gained a new function as a result of those mutations, but you can still tell that they're related to the original copy of that gene. Oh, and I had mentioned, well, I'd said that most mutations come from DNA pol III mistakes. There's also another source of mutation, which would be mutagens, which are just chemicals or physical factors in the environment that cause mutations to happen. An example would be a chemical that causes mutation, or it could be UV light. UV light will cause mutations as well. Anything that causes a mutation to occur that's like physically present in the environment would be a mutagen and is a potential source of mutations. But of course that's going to depend on what the microbe is exposed to in its environment. Then you have recombination. In recombination, you're not changing any of the DNA sequence, but you are kind of cutting it out of the genetic element that it's on and switching it out with a segment of a different genetic element. So in the diagram, this is actually showing recombination in a sexually reproducing eukaryote, such as yourself. So these would be chromosomes that are inside of one of your gamete cells, or just in the gamete cell of some eukaryote, some multicellular eukaryote that does sexual reproduction. And this gamete is preparing to divide, which means it's copied as DNA. So originally, this gamete is going to have this chromosome here, maybe from the mother, and this chromosome here, maybe from the father. And since it's preparing to divide, it's going to copy its DNA, replicate its DNA, so you get another copy of the mother's chromosome, another copy of the father's chromosome. And now, these chromosomes, if they're lined up together, which they would be if the cell is going to divide, they can exchange segments with each other. So here, a segment of the mother's chromosome has been exchanged with a segment of the father's chromosome. And a similar thing can happen in microbes. It wouldn't look the same, of course. Microbes are not going to have all those copies of chromosomes. They're just going to have their one circular chromosome, and then when they're dividing, they will copy it. So they'll have two circular chromosomes, and then they'll also have some little plasmids in there too. All of those things can have segments kind of cut out and replaced with a different segment by recombination. So there's not really any way to control where those cuts are happening. So it could be that whole genes are transferring, or it could be that parts of genes are transferring. So you can end up having new functions gained actually. If one part of a gene transfers and lines up with another part of a gene, and that just ends up being something that has a function. So now we'll go over the processes that drive evolution. First, natural selection. Natural selection can be defined as a process in which traits that are more advantageous are more likely to be passed down to offspring, so that over time those traits would become more common in the population, or the alleles for those traits would become more common in the population. This is basically guaranteed to happen as long as three assumptions are met, which are that different individuals in a population have different traits, or they have different alleles, so not everybody is the same. You also need to assume that those traits are heritable and they can be passed down from parent to offspring, which means that the traits need to be based on genes, since genes are what's passed down. And then finally, if you assume that some traits are more advantageous for survival and reproduction and some are less advantageous for survival and reproduction. So if those three assumptions are met, natural selection is guaranteed to occur. Basically, your individuals that have more advantageous traits are going to end up having more offspring, either because they're better at reproducing or because they just live longer and can have more children in a longer time period. Their children are going to get those advantageous traits. On the other hand, if you had an individual who had disadvantageous traits, they would probably have less children. Their children would also get those disadvantageous traits. So then in the generation of the children, you're going to have more children that have the advantageous traits and less children that have the disadvantageous traits. And this process will just repeat until eventually the advantageous traits become extremely common and the disadvantageous traits become so uncommon that they're basically non-existent, or maybe they're even completely eliminated from the population. At that point, we would say that this population has evolved. So when we say that a trait is advantageous or disadvantageous, we're talking about its effect on the individual's fitness, which can be defined as the individual's ability to produce viable offspring. So that means not just that they need to live, but they need to have children, and those children need to live to have children of their own. So if you have a child who is sterile or who just dies right away, that's not considered viable offspring. That doesn't contribute to your fitness level. If you don't have children at all, but you live to be, 200, you're not fit. In this sense, you have to have children or reproduce in order to be fit, and those children need to survive to have children of their own. So this is really what matters in natural selection. It's not how long you live, it's how many children you have, and out of those children, how many will be able to have children of their own. In this diagram, or in this kind of picture here, you see an example of natural selection. So here we have moths in two different environments. These moths are living in a forest that has dark trees, and then these moths live in a forest that has light trees, maybe aspen. So for these moths, they could either be dark in their color or they could be light. If these moths are in a population that lives in this forest with the dark trees, the dark moths blend in really well with those dark trees, but the light moths stick out very obviously. And then you're going to have birds in the forest who like to eat those moths. It's going to be easy for those birds to see the white moths, and it's going to be hard for them to see the dark moths. So that means that being white or being light colored in this environment is a disadvantageous trait. These light-colored moths are much more likely to be picked out and eaten by the birds than these dark-colored moths. Since these light-colored moths are not going to live as long, that means they're not as likely to have children, or they're likely to have less children than the dark-colored moths. Which means in the next generation of moths, you're going to have more that are dark-colored, because those are the guys that had all the children, and less that are light-colored, because those are the guys that weren't able to have a lot of children. If these moths move to a different forest where the trees have light bark, the situation will be different. Now the light-colored moths have an advantage. They blend in with the trees, and the dark-colored moths stick out really well, which means the birds are now going to be eating dark-colored moths more likely than the light-colored moths. Here in this environment, being dark-colored is a disadvantage. Being light-colored is an advantage. These light-colored moths will have more children. Dark-colored moths will have less children because they're being eaten more frequently. And over time, you're going to get fewer dark moths here and more light-colored moths. So some final points to make about natural selection are that while evolution acts on a population always, natural selection acts on individuals. So an individual will have traits that are advantageous or disadvantageous or neutral. An individual will have a certain number of viable offspring, but evolution is occurring only on the population level. Individuals do not evolve. They do not change their traits. They do not change their allele frequencies. It's the population as a whole that's going to evolve because of kind of the sum of all the effects that natural selection is having on the individuals within that population. And then finally, natural selection is not the source of genetic diversity. So it does not cause mutations to occur. It does not cause adaptations to occur. It only selects the ones that are good for that individual. It only selects the ones that are advantageous and will help the individual have more viable offspring. So mutations happen at random. There's no way to control whether or not you're going to get an advantageous mutation that would allow you to adapt to your environment. It's completely random. Once that random mutation has occurred, natural selection will act on it, and natural selection is not random. Natural selection is selecting only the mutations that are advantageous, and it's deselecting the mutations that are disadvantageous. So if you did not have mutation, then you wouldn't have evolution by natural selection. It would be impossible. Just because you have natural selection, that does not mean that you have mutation. Then the next process that has a large effect on microbial evolution is genetic drift, which is random changes in allele frequency or in trait frequency in a population. So natural selection is causing advantageous traits to be more common and disadvantageous traits to be less common. Genetic drift is just random changes. So it doesn't matter if the trait is advantageous or disadvantageous or neutral. It's just random. It's a completely random process, which means that changes that occur due to genetic drift are not going to be adaptive necessarily, which means they don't necessarily help individuals or populations thrive in their environment. They might be neutral, they might be adaptive, or they might have a bad effect. Actually, it is possible. They're completely random. So an example of genetic drift would be if you had this population of bugs. Some of them are red and some of them are blue, and they're all just kind of milling around there, and then someone comes to swat the bugs, and by random chance they swat all blue bugs. They don't swat any red bugs, only blue bugs. Then what do you have left? A lot more red bugs and a lot fewer blue bugs. So now if all the bugs have children, you're going to end up with even more red bugs and, a little bit more blue bugs. So this is genetic drift. We go from an equal population to a population that is skewed to red bugs simply because by random chance blue bugs happen to die more than red bugs did. In this diagram, it's showing kind of an experiment that you could actually do yourself. You just need a tube and you need little balls that are different colors. So in this original tube, you're going to have these 4 different colors of balls: orange, red, blue, and green, and they're equally distributed. So maybe you have 100 balls in there. 25 are orange, 25 red, 25 blue, 25 green. And you're going to basically select at random some of these balls and put them into another tube, into these 3 tubes. So your random selection of balls that went into this tube here just happened to have 2 oranges and no greens at all. So that means that the frequency of colors in this new tube is different from the frequency of colors in this original tube, but that's just because of a random process. It's because you randomly selected more orange and less green balls. Then if you again make a random selection of balls from this tube and put them in another tube, maybe you don't select any red balls at all this time. So now you have a population that's only blue and orange balls, just randomly. And then if you do a third selection, maybe you just so happen to only select orange balls, and so you end up with a population of entirely orange balls. And you've lost all the red, blue, and green. And it's not because red, blue, or green balls are disadvantageous. It's not because orange balls are advantageous. It's just because that's the way you randomly selected those balls. And then the same thing is happening in all these other tubes. And because every random selection is different, you end up with different final tubes, different ball compositions in the final tubes. So genetic drift is always affecting populations. The smaller the population is, the more likely it is to be affected by genetic drift. And that's one of the main reasons why small populations evolve more quickly than large populations. Right? If you have a very small population of these bugs, then when you swat only blue bugs, you just killed most of the blue bugs. There weren't that many to begin with because it's a small population. So now this population has, been significantly altered in its allele frequency or in its trait frequency because of that. But if this was just the little bit of the bug population that we're seeing, and secretly the whole screen is covered with these bugs, and you happen to swat a few of these orange bugs, or sorry, a few of these blue bugs, that's only a tiny effect on the total population of blue bugs, and so that's not going to have a big effect on the total ratio of blue bugs to red bugs in the population. So the smaller the population is, the more affected it will be by genetic drift. So now we'll talk about how evolution affects microbes specifically compared to other organisms. So microbes evolve much more quickly than any other organism, which means that when their environment changes, they're able to adapt to that change very quickly. There's a couple reasons for this. First, microbes have a very short generation time. It's usually measured in hours rather than like years for you. So of course, when you have generations occurring more quickly, you're just going to have evolution occurring more quickly. That's just, that's just an inevitable consequence of having shorter generations. But also, prokaryotic microbes are haploid, which means they only have one copy of every gene, just one allele every time for every gene. Eukaryotic microbes are diploid, so they have two copies, just like you. You have two copies of every gene. If you have two copies, you're going to evolve more slowly. If you have one copy, you're going to evolve more quickly. And that's because for organisms that have two copies of every gene, one of those alleles would be dominant and one would be recessive, or you would at least have two alleles for the gene, one dominant, one recessive in general. So in order to actually get the recessive trait, you need to have two copies of the recessive allele, not just one. If you have one copy of the dominant and one copy of the recessive, you're going to get the dominant trait, not the recessive trait. If you're a haploid organism and you got what would be a recessive allele, you're going to have that recessive trait. There is no way for it to be masked by another allele that's dominant over it. Even though microbes evolve very quickly, they speciate very slowly. Speciation is when you get an actual new species evolving from the original species. For microbes, it's actually really hard to even define what a species is because they reproduce asexually. But in general, we would define that based on their rRNA sequence in addition to maybe some other information about their traits or their genetics. So these rRNA sequences are changing very slowly. And so you get speciation for microbes very slowly. They can gain new traits quickly to adapt to their environment, or they can lose traits quickly to adapt to their environment. But in order for microbes to actually form a new species, it takes a long time. You also will have, for any given species of microbes, different strains that belong to that species that are all a bit different. And they actually can be very different on a genetic level when you're talking about their genomes. So for microbial species, we divide their genome into two pieces, the core genome and the pangenome. The core genome includes the genes that every strain that belongs to that species has, and the pangenome includes all of the genes that any of those strains have. So this Venn diagram here is showing you the core genome versus the pangenome. This would be a microbial species that has 3 different strains. So here you have the genome for strain 3, for strain 2, and for strain 1. And you can see that in here are the genes that all 3 of those strains have in common together. So all the strains of the species have these genes in the middle here. So this is the core genome. You have a lot of genes that only strain 1 has and strain 2 or 3 don't have. Then you have some genes that strain 1 and strain 3 have, but not strain 2, so on and so forth. All of the genes here all together are the pangenome. So everything here is the pangenome, and then only this part where all the strains overlap in the middle is the core genome. Another feature of microbial evolution is that they tend to lose genes very quickly if those genes are not being used, or if they're not really used frequently. And that means that microbes will have generally a very small genome compared to other organisms, which can have a very bloated or very large genome. This is probably because if you have two microbes kind of living together and both of them are able to do the same reaction, they both, let's say, need this reaction and both of them can do it. Maybe it's a reaction to create a particular amino acid. Both of them can do it, which means they have the genes for the enzymes that let them make that amino acid. But they're each making kind of more than what they need, so they end up excreting some of that amino acid outside of the cell, and then the nearby cell, the neighboring cell from a different species, can pick that amino acid up and use it. So that means if you have a mutation in this neighboring cell of a different species that means that it's not able to make that amino acid anymore, it's still going to be able to pick it up from the environment because its neighbor is making it still. And that means that now it doesn't need that gene at all. And that gene is probably going to be deleted from the genome by evolution. Which would mean that in the future, this microbe is going to be fine, but only if it has its neighbor there making that amino acid for it. If it doesn't have that neighbor making the amino acid, or some neighbor making that amino acid, it's not going to be able to make it itself because it lost those genes. So now it's dependent on neighboring species to supply it with what it needs. This is very common to find for microbes. They're very frequently living in communities with each other. You'll have different species, different strains living together in community, and they're each kind of bringing something to the relationship. So each is kind of doing something that the others can't do, but the others will all need. And they're all mutually dependent on each other. They cannot live without each other or without someone to fill those roles that they can't do anymore because they deleted those genes. So this graph is showing an experiment that's kind of illustrating how quickly genes are deleted from a genome if they're not used in microbes. These are going to be photosynthetic bacterial cells. You have two populations of them. You have a population that's grown in the light. So they're exposed to light and they're doing photosynthesis. Then you have another population of cells that's raised in the dark. That's shown by the blue line. So since they're in the dark, they don't do photosynthesis, even though they have all the genes for it. And if you watch those genes, in this experiment, they specifically watched a gene for pigment, and you can watch those genes vanish over time. So in the microbes that are living in the light, if you have a mutation in one of those microbes that it loses the pigment gene, that cell is not going to survive compared to the other cells. It's just not, well, without the pigment, it can't do photosynthesis. So it's not going to be making energy as much, it's not making glucose as much, it's just not at an advantage compared to the other cells. The other cells have a major advantage on it. So they're going to reproduce quickly, this guy reproduces very slowly, and these guys are going to be basically remaining very common in the population. Overall, this population of cells is going to be all pigmented cells because that pigment is useful to them since it lets them do photosynthesis. If you look at the population of cells that were raised in the dark, once they get that mutation, once one of them gets that mutation where the pigment is deleted, since nobody's doing photosynthesis here, this guy is actually not at a disadvantage at all. If anything, he's at an advantage because he's not making something he doesn't need. All these dudes are making a pigment that they can't even use since they're in the dark. And it costs energy to make things, so they're spending energy and getting nothing. Whereas this guy is not spending that energy for nothing. He's conserving his energy for things that are actually useful to him. So as time goes by, he's going to actually reproduce a little more quickly than the other cells in the population since he's at an advantage since he's not wasting energy. So you're going to have more and more cells that are descended from that guy that don't have the pigment gene, and as time passes, more and more cells won't have the pigment gene. Eventually, within just 20 generations, all of those cells basically do not have the pigment gene anymore. So that's a very brief amount of time, evolutionarily speaking, for a gene to be almost completely lost from a population just because it was no longer essential for growth. Okay, so now we'll go over systematics for microbes, which is kind of like how they're classified and related to each other. So a technical definition of systematics is that it is the study of diversity of life, focusing on the relationships between organisms. Within systematics or related to systematics, we also have phylogeny and taxonomy. Phylogeny is studying the evolutionary relationships between organisms. So modern systematics is all based on phylogeny. We're not interested in just which organisms have related traits. We're interested in which organisms are related to each other by common descent and share a recent common ancestor, or how organisms have evolved from each other, which is phylogeny. Then you also have taxonomy, which is the science of classifying organisms, basically putting them, into families and orders, and phyla, and then domains. So the phylogeny is always going to be studied using some molecule, some macromolecule that has a sequence that carries information. So of course that means DNA and RNA, but that can also mean protein as well. Actually, you could compare the amino acid sequence of proteins in different organisms that do the same function and see how many amino acids they have in common in that sequence, and that would tell you something about the probable evolutionary relationship between those organisms. But we're usually going to be using DNA actually for phylogeny studies. Specifically, the rRNA genes would what be is most commonly used. In taxonomy, you're classifying organisms. That used to be done based purely on physical characteristics. So if they look the same, they're classified the same. For kind of non-microbial life, we have gotten away from doing it that way. Classification is now based on genetics, on phylogenetic relationships. So we're trying to classify things in the same category if they're actually evolutionarily related to each other, like closely. So a genus should be organisms that are all really closely related to each other by evolution. For microbes, it's a little hard to base it entirely on phylogenetics because we don't know the genome for all of these different organisms. There's just so many of them, and because you have the horizontal gene transfer. So just because you see the same gene in two different organisms, it doesn't mean they got it from a common ancestor. It might be from a common ancestor, or it might be there from horizontal gene transfer. So for microbes, we use kind of a combination to classify them of their phenotype, which is their physical characteristics, such as like what they look like, if they have a color, what metabolic reactions they do. We would also base that on their genotype, which is the genetic sequences that they have, and we would base it on their phylogeny, which is their evolutionary relationships. And then pictured you have the universal tree of life, which is a phylogenetic tree, which means that it is showing how organisms are related to each other by evolution. So when you're doing a phylogenetic study, you're going to go through these kind of steps. First, you need a sample of DNA. If your microbes are cultured in the lab, then it's pretty simple to get a DNA sample from them. But if they're not cultured, it's more complicated because that means that maybe you have a sample of soil, you have a sample of water, but you don't know who all is in there. You just know there's going to be a lot of different species in that sample. So you can get a DNA sample from it, that's not a problem. But what is a problem is knowing whose DNA that is. It might be, from a variety of different species, and you're not going to know who all those species are, how many there are, what parts of the DNA are from which species. So it gets really complicated if you don't have those microbes growing in culture. After you have your DNA sample, you need to amplify it, basically making a lot of different copies of it. Usually you're going to get just a teensy amount of DNA from that sample, but you need a lot. So you're going to use some technique to copy it. And the technique that we use is PCR, or polymerase chain reaction, which we're going to go through later in the semester. Actually, towards the end, we'll talk a bit more about PCR. But for now, all you need to know is that that is a technique that is done to copy DNA. It makes a lot of copies of DNA. Then you're going to sequence the DNA, meaning you need to know exactly the sequence of nucleotides in that DNA sample that you have. There's a lot of different ways to do that, actually. Yeah, a lot of different techniques that you can use to do that. Some of them are kind of slow and labor intensive and older, but maybe cheaper. Some of them are kind of newer and super fast and efficient, but it requires really expensive machinery. When you're sequencing the DNA, you're usually not going to sequence literally the whole thing. You're probably going to have something in mind that you're looking for, and usually it's rRNA. Specifically, you would usually be looking at the sequence for the small subunit rRNA genes. Gene or genes. Some of them will have multiple, actually. Multiple rRNAs in the small subunit. This is actually kind of a 2D diagram of that small subunit rRNA. So you can see it's got quite a complicated structure. If you unfolded it, it would be very long. It has a lot of nucleotides in it. So you're going to be sequencing this. You want to know what's the exact sequence of nucleotides in this small subunit rRNA. You could also be using a different gene. It doesn't have to be the rRNA, but it usually is. Then you need to align the sequence and generate a tree. For both of those steps, happily, you're probably going to have a piece of software that's doing it for you. If not, you're going to be spending like a million years on it because it's real labor intensive. Aligning the sequence basically means, taking that sequence from your sample, comparing it to a different sequence from another organism, and making sure that it's lined up right with that other sequence. We'll go into the details there in a moment. Then generating the tree means, based on these sequences for all the organisms that you're comparing, what's the best way to say that they're related to each other? If you were going to draw a tree, a phylogenetic tree that shows their relationships, what's the simplest way to draw that tree based on these different sequences that you have? So your software would generally draw like a bunch of different trees and then pick the one that is the most logical or the most simple. Here you have kind of your workflow for this. You start with your bacteria or archaea, your microbes. They're growing. Hopefully they're growing in culture so you can get a clean DNA sample from them. So you're going to take a DNA sample from these microbes. Then you're going to amplify it by PCR, making lots of copies of the DNA. Actually, at this point, you're generally already going to have picked out what gene you're looking at, and you're going to be trying to make copies just of that gene, not of the rest. Whenever you're doing PCR, then you need to run it on a gel, basically loading this copied DNA into a gel, and then it'll migrate through the gel because it's being attracted by an electrical current. Actually, you guys did this in 1306. You ran some DNA on a gel in one of the labs. Then you would need to actually cut out the little bands, of DNA from that gel and purify it again. And then you can sequence it. And in this particular sequencing machine, you would get a graph like this that has peaks in different colors for A, C, G, and T. So you would see an A peak, a C peak, a GG peak, and a T peak. So that means the sequence is A, C, G, G, T. In general, now a computer software is going to do this for you and just give you the sequence at the end. And then you're going to have your software line the sequence up with another sequence from a different organism that's from the same gene. And then, well, actually, we'd probably be looking at multiple different organisms, to be honest. And then based on all that information, you're going to make a tree that is the most logical and most simple tree that you can make that shows the relationships between those organisms. And your software is going to be doing that too. Thank God. So one of the first and most important things in this process, or not the first, but one of the earliest things, and one of the most important things, is picking which gene you're going to be looking at. So you want to pick a gene that has a homologue in other organisms. So a gene where there's another gene for it, that's a homologous gene. Homologous genes are genes that have come from the same common ancestor. And we a lot of the time call them homologs because that's shorter. So you can't necessarily tell by looking if two genes are homologs, but once you sequence them, in general, if their sequences are very similar, they're probably homologs. So they're probably coming from the same common ancestor. But that's not 100% always true. Sometimes you might have two genes that are different. They do not come from a common ancestor and they are different from each other, but they have mutated so that they look like each other pretty closely just by random chance. That is not likely to happen, but that does happen. So most genes that have a similar sequence are homologs, but not all of them are. You have to kind of be aware of that possibility. There's a couple different types of homologous genes. You could have orthologs or paralogs, and we really want to pick an ortholog for our phylogenetic analysis. Orthologs are homologous genes that share the same function. Paralogs are homologous genes that have different functions, which means that they've evolved different functions over time. You want to stick with the orthologs generally, just because it's a clearer comparison. If you have two homologous genes that came from the same ancestor and are still doing the same function in these two different species that have them now, then it's a pretty straightforward comparison between the sequences of those different genes. For paralogs, since they have different functions, you're probably going to see that the sequences are more different. At least one of them is going to have gained a new function, and it's probably going to have a more significantly different sequence than orthologs would have from each other. So you want to find some type of ortholog that you're going to be analyzing for your phylogenetic study. Probably that ortholog is going to be the small subunit rRNA gene, but it might be something else as well. So here's a diagram showing the relationships of different genes to each other. This is for a histone gene, the H1 gene. So you have, at some point in the past, the histone H1 gene in a common ancestor for humans and chimps. Then you had a duplication mutation event, so you ended up with two copies of the histone H1 gene, and they started evolving differently. So here you have histone H1.1 and histone H1.2. So these are going to be homologous genes because they share a common ancestor. Then once humans split from chimps in this lineage, you're going to end up with the human H1.1 gene and the chimp H1.1 gene, and also a human H1.2 gene and a chimp H1.2 gene. So these guys are still all homologs of each other because they're all coming from this ancestral histone H1 gene. But now there's two different variants by a duplication mutation, and you have two different species, so that's double the variants. Now you have 4 different variants. Actually, the H1.1 genes have one function, and the H1.2 genes have evolved a different function. It's a related function, but it is a little different from the H1.1 function. So these H1.1 compared to the H1.2 genes, those are paralogs. They come from the same ancestral gene. They have a common ancestor, but they do not have the same function. So those are paralogs. In the chimp, they're also paralogs. H1.1 and H1.2 are paralogs. But if you compared the H1.1 in humans to the H1.1 in chimps, they have the same function still, and they share a common ancestor. So those are orthologs. Same thing for H1.2 in humans and H1.2 in chimps. Same function, those are orthologs. So you might actually pick those genes for your phylogenetic study. You might pick H1.2 in humans and in chimps, but you would not pick H1.2 in humans and H1.1 in chimps, because those would be paralogs, not orthologs. Okay, so once you have your gene picked out and you've amplified it by PCR and you've sequenced it, now it's time to align that sequence with the other orthologs from different species that you're looking at. All right, so you have your sample gene that's been sequenced, and then you're going to look in a database. There's actually publicly maintained and publicly available databases that have the sequences for genes that have been sequenced. So you're going to look at one of those databases to find the orthologs in different species for the gene that you're looking at. You're going to pull those sequences, and then you're going to align them with the sequence from your sample. The reason why you have to do this alignment is because you can have insertion mutations or deletion mutations that have changed the way those sequences line up. And in an insertion mutation, you've got one or more nucleotides that have been added into the sequence. In a deletion mutation, you've got one or more nucleotides that have been taken out of the sequence, and that's going to change how they're lining up. So in this example, here you've got maybe your sample on top. So this is the gene from your sample that you actually sequenced, and then here's two orthologs that you found the sequence of in a database. If you just line them up like this, this is not actually a correct alignment, because there's been some insertion mutations or some deletion mutations. So actually, these CCTs should be lining up together. And here you just have 3 C's, but that's lining up with these C's as well. That should all be lining up. So you're going to basically insert some blank spots between the GGA and the CCT so that the CCTs can line up. So this is what the sequences might look like after you've done that alignment, which means maybe these guys, the AAA and the GGG, those might be there from an insertion mutation, or those could have been there originally, and then you had deletion mutations and the other 2 orthologs, taking the AAA out and the GGG out. You actually would not necessarily be able to tell if it was an insertion or a deletion mutation just from looking at this alignment, although you can maybe make a guess. So after alignment, the sequences look much different from here, and they line up with each other much better, much better. So this is really important to do, so you can make sure you're comparing the sequences accurately. Here's another example of an alignment. This is actually not for DNA this time, this is for a protein sequence. We've got on the top the sequence of amino acids in the sample that they're studying, and then you have the sequence of amino acids in 3 proteins that are made from an ortholog gene, orthologous to this target, the gene that produces target protein. So these should all be the same, basically, but they've got some insertions and deletions, so they don't line up perfectly, and you need to align them so you can compare them accurately. And of course, when you're looking at these sequences, it's not going to be short like this. It's going to be long like this. And actually, it's probably going to be a lot longer than this. I cropped this picture, actually. Yeah, these things are usually extraordinarily large. The proteins and the genes that they come from are generally extraordinarily large. So if you were doing that by hand, you would be there just for days, pulling your hair out, trying to align all those things, like hundreds of different bases or hundreds of different amino acids. Even worse if it's a gene, because for every gene you're going to have 3 bases for each amino acid in the protein that it produces. Right, so if you have a gene that's 500 amino acids long, if you have a protein that's 500 amino acids long, the gene for it would be at least 1,500 nucleotides, which is a lot. That's a lot to do, but that's not a lot for a gene to have. So the algorithm is going to be doing this for you. You're going to have some software that is doing this for you, thank God. But at the end, you're also not going to know if it's correct. There's not a way for you to know if the alignment is done correctly. You just do the best you can, or the algorithm does the best that it can. So you're going to figure it's pretty right, but you're not going to know how right it is for sure. Once you've got your sequences aligned, then you can compare the differences between them. So if you would just compare all the times that maybe sequence 1 has a base that's different from sequence 2, we've got one difference right here, we've got another difference right here, and we've got a third difference right here. So that means we've got 3 differences between sequences 1 and 2. So you would do that for also comparing 2 and 3, and then comparing 1 and 3, and you can make a little table like this, or the computer will just have this stored in its memory. And then this information about the differences is what you're going to use later to construct the phylogenetic tree. When things have less differences, they're going to be closer together in general, phylogenetically. If you did not align the sequences before you did that, before you compared the differences, you would get way more differences, and that would not be accurate. Okay, so a little bit more on phylogenetic trees. They have nodes and branches. So a line on the tree is a branch, and then where you have a split, that's a node. Any place where you have a node, that means that you had speciation there. That's a speciation event between species 1 and species 3 here. This is their common ancestor species, and then it's speciated into species 1 and species 3. The length of the branches is showing in general how long ago that occurred, or how many mutations you had. Because I guess if you're just analyzing the sequence, you can try to figure out how long it takes for mutations to occur. That depends on kind of the gene that you're looking at specifically, but that's kind of more like an estimate. So you would know exactly how many mutations had occurred though, or how many differences there are. So the length of the branches is showing basically how many mutations occurred between, let's say, this common ancestor when it speciated into species 1 and 3, and when it speciated from its original, most common, even previous common ancestor. Basically, the longer the branch is, the longer the time has passed between speciation events. Then you can have trees that are rooted or trees that are unrooted. Usually we're used to looking at a rooted tree, which shows where the last common ancestor for everybody on the tree is. So this guy at the bottom, this is the root. This is the last common ancestor for every species on this tree. They all evolved from this guy. So usually when we see a phylogenetic tree, it has a root like that. But it actually doesn't have to have a root. You could have an unrooted tree where you don't show that last common ancestor. Sometimes you're not able to know like when that last common ancestor would have come about, or who exactly he would be related to most closely. So you might not be able to add a root to your tree depending on how much information you have. In order to build the tree, you're going to be first aligning your sequences, then comparing the differences between the different sequences. So you align your sequences. You would probably actually make an insertion here so these T's line up with this T. That would also cause this G to line up with these G's, so that would be a better alignment. Then you're going to make a table showing all the differences between your different orthologs that you looked at the sequence of. And then you're going to make a phylogenetic tree based on that, trying to group things together that have fewer differences between each other. So if you've got 4 differences between species 1 and 4, then they're probably not going to be right next to each other. But species 1 and 2 only have 2 differences, so they probably would be right next to each other. And in the final tree, that is correct. Species 1 and 2 are right next to each other. Species 1 and 4 are not, because they diverged earlier from each other. Here, the branch lengths are always 1, so you have like 1 difference here and another difference here. So when you had your common ancestor here, that was the most common ancestor for species 1 and 2, and that guy speciated into species 1 and 2. Each of these, species 1 and species 2, developed 1 mutation in this period of time. If the branch was twice as long for species 1, that would mean that you had 2 mutations there compared to only 1 in species 2. Whenever you're doing this, there's a lot of different ways that you could draw your tree. And you're not going to know which one is right. So when the software is doing this for you, it's going to draw a lot of different trees. And it's going to pick the one or the several that are the most correct looking, that are minimizing differences between the organisms it's showing as being closely related to the greatest extent possible, and just the tree that seems the most logical and the most simple. But you're never going to really know, is that the right tree? So we try to add more and more information, more and more sequences to get better and better trees. And you can also kind of do an estimate on which part of the tree is more correct and which part is less correct. But you're never going to know for sure that this tree is the most correct tree that's possible. And finally, we'll talk about horizontal gene transfer in more detail. So in horizontal gene transfer, you're transferring genes between individuals, but you're not doing that through heredity. So normally genes are transferred between individuals when a mother cell divides to form daughter cells, and the daughter cells have that same DNA from the mother cell. So they've gotten the genes transferred from the mother cell to them. That's called vertical gene transfer. That's usually how genes are transferred. But for microbes, they can also be transferred horizontally between organisms that are not actually related to each other by heredity. So we had mentioned that plasmids could be shared through a sex pilus by different bacteria. And so that is one method of horizontal gene transfer. So you could have two bacteria next to each other that are not related, that aren't even part of the same species, and they could share genes with each other through a sex pilus, potentially. Usually you share a plasmid, but occasionally you could actually share a whole chromosome or part of a chromosome. There's also a couple other mechanisms. So here you see the different mechanisms. This is conjugation, where genes are shared through the sex pilus, and you don't need to know the names of these methods, but here's plasmids being shared through the sex pilus, so that's one way to do it. Another way is that when a bacterial cell dies or an archaeal cell dies, it's going to end up releasing its fragmented DNA. The DNA will be fragmented, and then that just ends up lying around in the environment. Other bacteria will sometimes pick that up. They'll find the little fragments and they'll pick them up. So now they have those genes. And then the third way involves viruses. Some viruses will actually inject their DNA into the host cell. That DNA will insert itself into the chromosome of the host, and then later on it'll be removed and packaged up into a new viral particle. And sometimes when that happens, it doesn't get cut out right, so you actually take part of the host cell chromosome 2, and then this virus or the new virus would end up with the viral DNA and also part of the host cell's DNA, and then it's going to go to a new host and infect that host and inject that host with the DNA, and so the new host would then get the DNA from the previous host. So this actually happens in microbes, but it also can happen in larger organisms, including you and me. So some of our DNA in our genome has actually been introduced to the human lineage by viruses in this manner. It's estimated that maybe 8 to 11% of our DNA is actually coming from viruses, and would have been therefore horizontally transferred to us. But it's not very common for multicelled life forms that are large. It's more common in microbes and specifically in prokaryotes, not even in eukaryotic microbes. So in eukaryotic microbes, you could have genes shared horizontally using this method, but eukaryotes will never do the sharing plasmids through a sex pilus, and they'll never do picking up little fragments of DNA they happen to find lying around. That's only for prokaryotes. So prokaryotes are going to have the most horizontal gene transfer. In contrast, here you have normal vertical transfer of genes, where you have a mother cell that divides to form two daughter cells, and they have the DNA from the mother cell. So that's regular. So we're assuming that when we see two different species of microbe that have the same gene or have an ortholog, share an ortholog, we're assuming those orthologs have come from the same place, and that means that those species have come from, they've evolved from a common ancestor, or they share a common ancestor, and they both have that same gene because it's been transferred to them vertically through natural heredity, or, well, horizontal gene transfer is also natural, through standard heredity. But it's also possible that that gene is there in one of the species because of heredity, and it was actually horizontally transferred to the other species. So you can't just assume necessarily for microbes, especially for prokaryotes, that because they have the same gene, they share a common ancestor. That makes it way harder to tell how those microbes are related to each other. Really, really hard. There's some cases where you're able to detect when horizontal gene transfer has occurred. There's also some genes that are not generally transferred by horizontal gene transfer. And when we're building phylogenetic trees, we like to focus on analyzing those genes that are not commonly transferred horizontally, so that we can build an accurate tree and not have it muddied by this horizontal gene transfer. So rRNA is an example of a gene that is not commonly transferred by horizontal gene transfer. If you were to draw a completely accurate tree of life that shows the transfer of genes horizontally, it might look something like this. In the very early stages of life, you had a huge amount of horizontal gene transfer between the cells. We'll probably never actually be able to work out the exact relationships evolutionarily between these guys, partly because they're long dead, and partly because of all the horizontal gene transfer that was occurring at that time. Once you had eukaryotes emerge, we didn't really have any more horizontal gene transfer for eukaryotes. You know, it can happen by viruses, but it's just not very common. But for bacteria and archaea, the prokaryotes, you've continued to have horizontal gene transfer, playing a role in their evolution, like a significant role. And of course, endosymbiosis can be seen as a form of horizontal gene transmission, where mitochondrial DNA in eukaryotes is actually coming from proteobacteria, and chloroplast DNA in eukaryotes is actually coming from cyanobacteria. And it says plastids here because that's like the type of organelle that a chloroplast is. Horizontal gene transfer, as a final note, is actually one of the reasons why we're probably never going to be able to put viruses on the tree of life accurately, because they actually do a huge amount of horizontal gene transfer. Some of their genes are from different viruses, some of them are from like bacteria or archaea or from eukaryotes. They're all the time doing horizontal gene transfer, which makes it just very difficult to impossible to know where they really are coming from evolutionarily. So next we'll talk about microbial species. It might seem like it's pretty straightforward to know what a species is, but for microbes it really isn't. In general, the definition of a species is that it's similar individuals that interbreed with each other and produce viable offspring. Viable offspring meaning offspring that are fertile and can have children. But these individuals could not produce viable offspring if they bred with members of another species. So as an example, you can actually have a horse breed with a donkey and their offspring is a mule. But the mule is not considered viable offspring because mules are sterile. They will never have offspring of their own. So that means that horses and donkeys cannot interbreed to produce viable offspring, and they belong to different species. The other, somewhat less common example of that would be lions and tigers, which can interbreed to produce a liger. But ligers are also sterile, so lions and tigers are different species. So this definition of what is a species is entirely based on sexual reproduction, right? Individuals breeding with each other, that is sexual reproduction. But microbes don't do sexual reproduction with just a few exceptions. Like all prokaryotes do not do sexual reproduction. So we need to have a completely different definition for what is a species for microbes than for things that are not microbes. Actually, we don't have one. There is no universally accepted definition for what is a microbial species, but we have some definitions that are kind of commonly agreed on by a lot of scientists. The most commonly agreed on definition for a microbial species is that it is a group of strains that have similar genetics and similar characteristics and that share a recent common ancestor. So that's based on genotype, similar genetics, that's based on phenotype, similar characteristics, and that's based on phylogenetics, which would be the recent common ancestor that they need to share. You can pick different genes to analyze to determine the genetics and how similar they are, and to determine when's their most recent common ancestor for a couple of different strains. We like to pick rRNA genes. We usually pick multiple genes actually, rRNA genes being just one of them, but in general, rRNA genes have like a heavy weighting in determining if two strains belong to the same species or not. And we have kind of a rough cutoff of 3%. If the rRNA genes of one strain are less than 3% different from the rRNA genes of a different strain, we might say that they're the same species. If they're more than 3% different, then we might say that they're different species. But it doesn't have to be rRNA genes that you're looking at. It could be actually any ortholog. So when you're looking at other orthologs other than rRNA genes, the cutoff we use is usually 5%. So if you have more than 5% difference in a pair of orthologs between two different strains, then they're probably belonging to different species. If you have less than a 5% difference, they're probably belonging to the same species. Then we would also look at their characteristics. That includes, especially for archaea, what fatty acids do they have in their membranes. It also includes what metabolic reactions the cells can do. Any characteristic that isn't too hard to analyze actually could be used to determine if they're the same or a different species. So in this diagram here, we're looking at a population of different strains. Every little circle here represents a different strain of microbe, and we're kind of grouping them into species. So every kind of green circle here that encloses some of these little strains is enclosing a species. So you would have in this species, all 5 of these strains are part of the species. For this species, you've got also 5 different strains actually, but 2 of them are almost on top of each other here. So those are all a species, which would mean probably their rRNA genes are less than 3% different. And maybe they do similar metabolic reactions and have similar components of their membranes. Here for species B, this is 6 different strains that are all belonging to that species. And then in yellow, these are other strains that we have not yet classified and determined what species they belong to. This isn't like kind of a straightforward determination necessarily. So this diagram here is showing that. Here's your type strain, which is actually the strain that is kind of used to define the species in the first place. And not because it's the most typical of the species actually, but because it's the first that was described. The first strain for the species that was described is the type strain for that species. And then you're just basically kind of trying to see if other strains are close enough to that type strain to be part of the same species or not. So if you're looking at strain B and strain A, and you're trying to decide, are they sufficiently similar to the type strain to be the same species as the type strain, maybe you look at their different orthologs. So in this case, we've done an analysis of the ortholog genes for these 2 strains versus the type strain to see how similar they are overall. So the ortho-ani score is basically the overall similarity in the genetic sequence between each of these strains and the type strain. So strain B has 94.9% genetic similarity to the type strain in terms of its orthologs. Strain A has 95.1% genetic similarity to the type strain in terms of its orthologs, which would mean that strain B is like 5.1% different from the type strain. Strain A is 4.9% different from the type strain, which means that if we're just going by this 5% cutoff, then strain B would not be in this species, and strain A would be considered the same species as a type strain. But actually, we're considering strain B to be part of this species, and strain A is not. And that's because there's other considerations that are going into this analysis. So we're also looking at the characteristics. Maybe strain B shares the same membrane fatty acids as the type strain, and strain A does not. Maybe strain A does some reactions that the type strain doesn't do, but strain B does do those reactions. So it's kind of nebulous whether 2 strains are going to be considered together in the same species or not. And the opinion on that can change throughout time, and it does. And, it gets kind of complicated and, honestly kind of frustrating to determine if strains belong to the same species or not. Okay, now for taxonomy. In taxonomy, we're classifying organisms into groups that start small and then get bigger and bigger. Each of these groups is called a taxon, or altogether they're called taxa. At the smallest end of this hierarchy, you have the species itself, which for microbes would include a number of strains. At the opposite end, at the biggest end of this hierarchy, maybe you have life itself, or maybe you have domains. The largest taxon is the domain. So species, a group of species that are all closely related to each other, would belong together in a genus. A group of genera, because actually the plural of genus is genera, a group of genera that are all closely related to each other would belong together in a family. A group of families that are all related to each other belong together in an order. A group of orders that are related to each other would belong together in a class. A group of classes that belong together or that are related to each other would go together into a phylum. For microbes, actually, we don't have the kingdom. I should say for prokaryotes, we don't have the kingdom. For eukaryotes, we do use the kingdom taxon. But for prokaryotes, we would just skip from phylum to domain. So a group of phyla that go together would belong to a domain. So all of the bacterial phyla are in the domain bacteria. All of the archaeal phyla are in the domain archaea, and all of the eukaryotic phyla are in the domain eukarya. So that's the basic scheme for taxonomy for microbes, especially for prokaryotes. In general, it might be good enough to just know what the species is for a given microbe, but actually sometimes that's not fine enough detail because strains can have significantly different characteristics sometimes that are important. And a good example of that is E. coli. So everybody has E. coli living in their gut, and it's fine for it to be there in general. Most strains of E. coli are not dangerous for us, but whenever you hear about an outbreak of E. coli, they're not talking about just E. coli in general, any strain at all. They're talking about a specific strain, the 0157H7 strain that causes serious disease and can actually kill people. So most strains of E. coli are safe, but the 0157H7 strain is quite dangerous. So that means when you're identifying a microbe, a lot of the time it's good enough if you can identify what species it is, but for some things like E. coli, you really want to know what strain it is too, so you can know if it's going to be dangerous for you or not. Taxonomy sounds kind of tedious and maybe stupid, maybe semantic, but it's actually very important because it allows scientists to communicate with each other around the world. So scientists that are working in completely different countries and maybe don't even speak the same language can still communicate accurately about which microbes specifically they're studying and which ones they're talking about. This ends up being really important. It's more important than you might suspect at first. So when I was doing my doctoral research, I was studying neuroanatomy, which is the anatomy of the brain, and I was looking at the rat brain. And there's actually different naming systems for neuroanatomy. There's not one agreed upon taxonomy that we're all using. We have different naming systems. So some scientists will use one system for naming different structures in the rat brain. Some scientists will use a different system for naming structures in the rat brain. And they're not corresponding to each other necessarily one-to-one. There's some structures that exist in one naming system that don't exist in another naming system. And you can't tell just looking at the name of a structure in one of these systems if it's going to even exist in the other system or what structure it would correspond to. So I was actually trying to reconcile those different naming systems with each other and kind of compare maps based on those different naming systems with each other. And I can't even tell you how difficult it was to try and figure out if a structure in system 1 is the same as a different structure in system B or in system 2. It would be really nice if we would have just one system for naming structures. It's actually really important. I guess the final point in taxonomy is that when we're naming an organism, we'll use its species name and its genus name in a format that's called binomial nomenclature. So we will say the genus name first, and then the species name. The genus name will be capitalized, and then the whole thing will be in italics. So here you have Homo sapiens, that's us. Homo is our genus, sapiens is our species. Homo capitalized, the whole thing in italics. Here is E. coli. Escherichia coli is the full name for that. Escherichia is the genus, coli is the species. In order to actually name it, though, you would use both the genus and the species name, and you would say Escherichia coli or E. coli for short. So we're going to conclude with some of the official resources that are used to kind of standardize taxonomy for microbes. So first you have the International Committee on the Systematics of Prokaryotes, which is the ISCP. which is basically in charge of taxonomy for prokaryotes and publishes rules for naming them and a journal in which new discoveries related to taxonomy or phylogeny are published. So one of those things is the International Code of Nomenclature of Prokaryotes, or the ICNP, or just the Prokaryotic Code. So that's published by the ISCP. So that International Code of Nomenclature is just basically a set of rules for how to name a new prokaryote. If you are going to be naming a new prokaryote, not only do you need to follow those rules to name it, but you also need to be able to culture the type strain in the lab and deposit frozen samples of the culture into 2 different national collections. They need to be in 2 different nations, so that way that it's like internationally stored. If you're not able to culture it and deposit samples into 2 different collections, you have to put the word candidatus in front of the name. And that's to show that it's not for sure a new species yet, it's provisional. It's a candidate for species. It's a new candidate for being a new species. Some microbes are really hard to culture actually because they have very specific growth requirements, like really high salts or no water or no oxygen or a weird pH. Some of them actually would need like an extreme pressure. Some of them can't survive if they don't have like their symbiote living with them in the same culture. So for some microbes, it's really hard to actually culture them and deposit them into 2 collections, and they would be named with candidatus. The book doesn't talk about the prokaryotic code, it talks about the bacteriological code. So this code, this system of rules, used to be only for bacteria, but now they've expanded to include archaea, so they call it the prokaryotic code now. And then the journal that's published by the ISCP is the International Journal of Systematic and Evolutionary Microbiology, or IGSEM for short, IGSEM I guess. This is the journal where if you have described like a new species of prokaryote, you're going to publish there. If you have like a new phylogenetic tree for prokaryotes that you made, you're going to publish it there. So all the information related to taxonomy and systematics for prokaryotes that is new is going to get published in the IGSEM. They actually don't just publish stuff for prokaryotes, they'll publish stuff for eukaryotes that are microbes as well. Then you have the list of prokaryotic names with standing in nomenclature, or the LPSN, which is a list of all the species of microbes, well, of prokaryotes actually, that have been named so far in every phylum, in every class, in every order, in every family, and what their names are. So if you're just wanting to know what the names of different things are, you would want to go look in that list of prokaryotic names with standing in nomenclature. That's the official name endorsed by the ISCP for each described prokaryote so far. The book also mentions another database, the Prokaryotic Nomenclature Up-to-Date database. That's no longer active. It actually was merged with the list of prokaryotic names with standing in nomenclature earlier this year. So now it's just one resource that has all the names. And then, after all that, there's 2 big reference works for prokaryotes. Not only would those works have the name of the prokaryote, but they're also going to have like the full classification of it and the characteristics of it, like what type of metabolic reactions it can do and stuff like that. It has information for how you could identify this specific species of microbe, and it has like instructions on how to culture it, like if it has any special requirements that it needs. Basically, a bunch of information about each type of prokaryote or each prokaryotic species is found in these reference works. So those are Berge's Manual of Systematics of Archaea and Bacteria, and the Prokaryotes, a Handbook on the Biology of Bacteria. Some of that information is available online. Some of it you have to pay for a subscription for. And wow, does it get expensive. It's really, I guess, only for professionals. Only professionals are who they're expecting to like pay all that money to subscribe to these reference works.